All right, so today we're going to move into section 1.2 and we are going to start to look at the cell. I know in junior high you did look at the cell. You looked at some cell structures. Everybody knows the mitochondria, right? The mitochondria, and it doesn't matter whether I'm teaching Science 10 or Bio 30. Other organelles people are fuzzy on and forget, but the mitochondria, I think because it's got a fancy nickname, right? The powerhouse of the cell. Everybody remembers that one. But we're going to look again at those organelles. We're going to look at the cell membrane, the cell wall, differences between plant and animal cells. But before we get there, we are going to look a little bit about how we got to our understanding of the cell. So obviously, microscopes played a huge role, right? Robert Hooke, he coined the term the cell, although he really didn't know much about what it was. He just said, "Oh, look, a plant is made of these little chambers." Reminded him of a monk, where a monk would sleep, which were called cells. So he just named it the cell, right? But how did we get to our current understanding of the cell? Before we get there, I just want to review the scientific method real quickly because we are going to look at some experiments that were done in the past, and some of them weren't all that good. There were problems with them. So, just quickly, I want to review the scientific method. We did look at the scientific method early on in the course. Now, the scientific method starts, all science is done the same way, by the way, okay? There's this there's some people that tout this thing called the creation science. It's called. There's no such thing as creation science. There's a view out there that the Earth is 3,000 years old, that it was created by God, that everything on Earth was created exactly the way it is today, et cetera, et cetera. To put the tag science on it, it's not right. When we do science, science is done using the scientific method. Okay, it always starts with an observation. So we observe something about the world around us. Okay, we state a problem. From there, we identify variables. We formulate a hypothesis. Okay, now remember, you guys, a hypothesis is a plausible explanation. Okay, it's not, it's rooted in science. Then an experiment is designed, data is collected, and a conclusion is drawn. Now, here's where science differs from, just kind of coming up with a conspiracy theory or coming up with some kind of. Remember, guys, a theory in science is very different. So once a conclusion is drawn, then usually we go back to step 4. And this can be done over and over and over and over again. Many times, once a conclusion is drawn in science, these conclusions go out to the scientific community, and other scientists try to replicate those experiments. And usually there are flaws in those experiments, so we have to go back to the drawing board. And this can be done over and over and over again. And that's the strength of science. Okay, scientists are constantly trying to pick apart theories, right? And if a theory is really good and it's really grounded in science, then it sticks around, okay, until we maybe get more information or better technology or whatever. So science is always, always, always looping back. Now, if you will recall, there were a number of different types of variables in scientific experiments. There are controlled variables. So these are conditions that are held constant throughout the experiment. The manipulated variable, this is the variable that is deliberately changed by the experimenter. Sometimes the manipulated variable is also known as the independent variable. Okay, and the last variable is called the responding variable. Okay, and the responding variable is dependent on the thing that you're changing. Okay, so the responding variable is the variable that changes in response to the manipulated variable. Now, sometimes in experiments, not always, but sometimes we have what's known as a controlled group. And this is a group of participants in an experiment in which the conditions are not changed in any way. Sometimes there's a word that's associated with this, and it's known as the placebo. Okay, and the placebo is something that's given in place of the manipulated variable. So, for example, if you were trying to test the efficacy, so how effective a certain drug is, you would have two groups of people. You would have an experimental group; those are the people you would give the drug to, and then you would have a control group, and you would give them a pill, but the pill might be filled with sugar, right? And they don't know that though. So they think they're taking the pill, and that's known as a placebo. And we do that to see if the effect is actually because of the pill, or is it because people are thinking they're getting the pill, right? So let's just do a little bit of practice here, just to kind of refresh our memories a little bit about scientific method. In each of the experiments described below, identify the manipulated, responding, and a couple of controlled variables. So scenario number one, a science student wants to know if the amount of water given to a plant affects how tall it will grow. So what is the manipulated variable in that little experiment? Well, it is the amount of water given, right? That's the thing the experimenter is changing. What responds to the amount of water given? Well, how tall a plant will grow. So that's the dependent or responding variable. Now, what are some controls? Well, the type of plant used, the type of soil used, the amount of light that the plants are exposed to, the amount of fertilizer, et cetera, et cetera, et cetera, right? Everything else has to be kept the same in that experiment. Okay, so the amount of water, this is manipulated. And how tall it will grow, this is responding. Okay, and then there's lots of controls in there. Everything else is controlled. A pharmaceutical company wants to know if a new drug is effective in treating migraines. So, what is the manipulated variable? It's the drug that's being given. Okay, this is M, manipulated. What is the responding variable? Well, whether people feel better or not, and whether their migraines are less painful. Okay, that's would be the responding. Now, in this case, you might not have, well, you're going to have some controlled variables. Maybe, you're dealing with people that have no other underlying factors. You're dealing maybe with the same age group. Maybe you're telling them not to take any other medication. But in a case like this, it would be more common to have a control group rather than controlled variables. So you would give one group the drug, and the other group would get a placebo. Okay, scenario number three. A car company wants to know if the type of brake pads in a car affects the stopping distance. So what would be the manipulated variable? The, the type of brake pads, right on. And the responding variable would be the stopping distance. Now, what are some controls in here? Well, the cars have to move at the same speed, right? It would have to be on the same type of road. So you can't have one road that's full of ice and one road that's completely dry. Maybe the same type of car, okay, the same weight in each car, et cetera, et cetera. So you have to understand, ladies and gentlemen, that it wasn't that long ago that our knowledge of the living world was quite limited. Now, again, humans, we only live for around, 80 years, right? So for us, 80 years is a long time. But in a historical sense, it's not all that long. I had a grandmother who died a few years ago, and she was 101 years old. And I did a little speech at her funeral, And I said to the people that were gathered there, like, think about that. Think about what we've seen in her lifetime, right? We've seen 2 world wars. We've seen, vaccines come on to play in the 1950s, right? We've seen, the computer generation. We've seen, all sorts of things. Like 100 years in the life of a human is a long time, but in history, it's not that long. And it wasn't that much more than 100 years ago that we didn't know that there were things smaller than what we could see. We didn't know that there were microbes. We didn't know that living things were made of cells, right? People didn't know where things came from. There was a prevailing theory, and I'm using air quotes right now because we know that's not the proper way to use the term, right? But people thought in the old days that living things just miraculously appeared, right? And this was known as spontaneous generation. So spontaneous means just out of the blue, just out of nowhere, generation. A living thing was made. So there was a thought, believe it or not, this was an actual, people actually thought this. Now we laugh today because we know about cells, but back then they thought if you took some grain, you took some dirty underwear, and you put them together, that's how mice were created. Yeah, exactly, right? Not that they came from other mice. It's that, yeah, you just mix together some dirty underwear and some grain, and a few days later, poof, mice would appear. They thought that you got sick because somebody put a curse on you, right? Or because you were a bad person. They didn't know that there were germs, there were bacteria or viruses or fungi that caused these sicknesses, right? So they thought that you just spontaneously got sick for no reason. So again, it took scientists, or well, yeah, it took scientists, people that were curious about the world around them, to kind of say, you know what, maybe there's more to this. Now, this is a little more than 100 years ago, but this is kind of the first attempt to disprove this idea of spontaneous generation. And it was done by an Italian scientist by the name of Francesco Redi. And he set out to disprove spontaneous generation. So at the time, people thought that maggots were produced by rotting meat and air mixing together. So almost like, we just finished chemistry, almost like writing a chemical formula, right? Oxygen plus hydrogen equals water. They would say rotting meat plus air equals maggots. So Francesco Redi, he thought, you know what, nah, I don't think this is quite true. So he set out to disprove this. And he set out 3 jars. In jar number 1, he just had a piece of rotting meat just open to the air. Jar number 2, he placed a piece of rotting meat into a clear container, and he put a cork on top and sealed it. And then in jar number 3, he left the meat open to the air because, remember, they thought air was part of the equation. And he put a very fine piece of like cheesecloth on top so that flies actually couldn't get in. Okay. And what he found is that only the piece of meat that was completely open started to get maggots. And he deduced that it wasn't just spontaneous generation, it was actually flies that would lay eggs on the meat, and those eggs would hatch into maggots. Now, although this was a great step in dispelling spontaneous generation, you have to remember, this was a prevailing idea for probably 1,000 years or 2,000 years, right? So this idea was ingrained in people, and people don't like change, right? So even though he set out to prove, hey, look, I left this piece of rotting meat open to the air. All I did was prevent flies from getting to it, and lo and behold, no maggots. People were like, no, 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 you didn't disprove that spontaneous generation occurs. You just disproved that it occurs in larger animals. It still occurs in microorganisms. Okay, so people were like, well, you have this soup, and the soup is perfect, and you leave it out on the counter, and even if you put a mesh over the soup will spoil. It gets all cloudy and it gets moldy, right? So that doesn't disprove that spontaneous generation doesn't happen in microorganisms. Okay. Fast forward about 100 years, an English scientist by the name of John Needham, he's like, wait a minute, nope, there's got to be something there. If it's not happening in maggots and mice, spontaneous generation that is, then it probably doesn't happen in microorganisms. So he set out to disprove spontaneous generation in microorganisms. So what he did is he took some broth. So broth is full of nutrient, okay? So it's really good to grow stuff like mold and bacteria in because it's got lots of nutrients for the microorganisms to eat. So what he did is he took this broth and he boiled it, okay? Now, people knew that if you boiled water, it prevented people from getting sick. Okay, so that was known at the time. They didn't really know why. They just knew, okay, if you boiled water, let it cool, and drank it, you wouldn't get sick. But if you drank the dirty river water, you would get things like, typhoid fever and stuff. Okay, so anyway, they knew that boiling liquids prevents people from getting sick. So he took this chicken broth, he boiled it, okay, and then he sealed the flask. Now, initially, it stayed clear. Okay, it didn't grow mold, it didn't grow bacteria. But the problem is that Needham didn't boil it for very long. He just boiled it for a very short amount of time. And when he sealed the flask, he didn't seal it very well, right? There was still air in the flask. So when he sealed it, any microorganisms that were still in the air were able to get in the broth. And sure enough, after a while, it became cloudy and moldy. Okay, so he tried, but he failed. Now you have to remember that these scientists aren't doing these experiments in isolation, right? They're publishing their work. People talk. Europe is a small place. So word of Needham's experiment got back to Italy, and there was another Italian, Lazzaro Spallanzani, and he believed that microbes existed in the air. So he said, oh, Needham didn't get rid of the air, so those microbes were left behind. So he tried to improve on Needham's experiment. He boiled the broth for much longer, made sure that he totally killed all the microbes in the broth, and then he completely sealed the flask in a vacuum. Okay, he was able to pull the air out, completely sealing the flask with wax so that no air was able to enter. And sure enough, the broth didn't spoil. Okay, so you might think, great, he disproved spontaneous generation, right? But of course, there were the naysayers that said, no. There's a special life force in air. And by taking the air out of there, that air is needed for the magical spontaneous generation to take place. So no, you didn't disprove spontaneous generation. You just proved that you need air for spontaneous generation. Okay, so again, his excellent experiment was kind of thrown out because people just don't want to change. Fast forward another 100 years, and a French scientist by the name of Louis Pasteur comes on the scene. You guys may be familiar with Louis Pasteur if you've heard of the term pasteurization. It's named after him, right? So our milk is pasteurized, right? Meaning that they take our milk and they heat it up to kill microbes. So that's pasteurization. That's kind of one of the things he's famous for. The other thing he's actually famous for, and many people don't know, is that he finally put the nail in the coffin for spontaneous generation. Okay, and the way he did this was he invented a special swan-necked flask. So it's named after a swan. If you've ever seen a swan has these very elegant necks and it's got a curve in it. So he was able to produce this flask that had this bend in it. Okay, so why is this a benefit? Well, what he did is he put some broth in the flask. Okay, he heated up the broth, boiled it, made sure that all the microbes were killed. Okay, just like the last guy did, right? But what was the difference between Pasteur and Spallanzani? Well, Pasteur left the boiled broth open to air, but the air had to travel through the flask. So as the air traveled through the flask down the gooseneck, all the microbes and all the spores from the fungus and all the bacteria and everything collected in the bottom of that flask. So the air was able to pass into the flask. So air was able to get in there, but all the yucky stuff, all the microbes were trapped in the bottom of the flask. Okay? So now people couldn't say, oh, you didn't let air in there. He let air in there, and yet there was no production of microbes. And even to this day, I think, you might have to fact check me on this, but I think his original broth and flask is in a museum somewhere, and it still has not gone bad. It's still, there's no microbes in it. Okay? Now, just to put a cap on this, of course, he would have done this multiple times, right? Had multiple tries. But what he did is he took the broth a few days later. It hasn't gone bad. He tipped the broth, allowed the broth to go into that little curve, and then tipped it back. Guess what happened a few days later? We had microbes growing, right? So he proved that microbes were not the air, it was the microbes in the air that caused the spoilage. Okay, so here's another look at this. So here's his swan-necked flask. Okay, the curve prevented contamination, but as soon as you took that curve out and let the air just get in, you had contamination. Also, when he tipped the flask and picked up whatever was caught in that that groove there and tipped it back, then he got contamination. So he proved that the microbes existed in the air, we just couldn't see them. Now remember Robert Brown? Robert Brown had a really good microscope and he was able to see the cells. So Pasteur, all of a sudden, Pasteur's like, hey, look, there's these microbes in the air. I trapped the microbes in my flask. And other scientists are like, what are these microbes floating around all over the place? So people started to use microscopes, people like Robert Hooke, to start to look more closely at the world around them, right? You had Robert Hooke, you had Van Leeuwenhoek, who was looking at pond water and pollen and sperm and all these things, right? And they're finding these little tiny structures that you can't see with the naked eye. So Robert Hooke coined the term the cell. He was also the first one to see the nucleus inside the cell. Now, two other scientists, Schleiden and Schwann. M.J. Schleiden, he observed that all plants are made of cells, all plants. Not just the cork that Brown was looking at, but all plants were made of cells. And he found that the nucleus was responsible for directing the growth of other cell processes. And then Theodore Schwann, he came along and said that not only are plants made of cells, but animals are also made of cells. If you take Biology 30, in Bio 30, when we talk about the nervous system, we talk about these special cells called Schwann cells that are actually named after Theodore Schwann. So Schleiden and Schwann, and then a third scientist named Virchow, using their observations, came up with what's known as the modern cell theory. And this is again, more than 100 years later, we are still prescribing to this cell theory. And the cell theory has 3 points to it. It says that all living things are composed of one or more cells. So there are creatures that are made of just one cell, but it's a cell. Okay, a human being, it's estimated that we have anywhere from 10 to 100 trillion cells. But we're still made of a cell or cells. The cell is the basic unit of all life. And here's the real kicker that came from all of those scientists that we just talked about, ending in Pasteur. All cells are produced from pre-existing cells. There is no such thing as spontaneous generation. Now, some of you guys might be thinking in your head, well, what about the very first cell? Right? If all cells come from pre-existing cells, what about the very first cell? Well, in Bio 20 AP, we actually look at some theories on the origin of life. Okay, so you'll have to wait till Bio 20 AP. All right, so page 12 and 13 in your note package, there are a few questions that I would like you to answer, as well as making sure that you're reading along in your textbook. All right, get to it.